Hello everybody, my name's Kate Phillips and I'd like to welcome you to our special event about the Lord Howe Island stick insect. I begin tonight by acknowledging the traditional owners of the land that we're on today, the Boonwurrung and Woiwurrung people of the Kulin Nation, and I'm paying my respects to elders both past and present and to those young leaders who are now emerging. Tonight we are live streaming the lecture and so I'd also like to welcome those of you who are watching online. We have a fabulous lineup tonight, starting with the beautiful Lord Howe Island. We've got a film and then two great speakers and then time for questions from you in the audience. And you might have already seen the stars of the show, our live insects, live stick insects, which we breed here at Museums Victoria. So this is very much a story about a whole lot of different organisations that have been working together to conserve this species. After our film and speakers, there'll be time for questions. If you're watching on the live stream and you'd like to send us a question, on Twitter, our handle is @lecturesmv, and then you can send the questions with a hashtag lecturesmv. And so in that way, you can join in. So we're going to start with our film. This film was made by a young filmmaker, Asher Flatt, and Asher will be joining us from Bristol after the film, because he is now working at the Natural History Unit at the BBC. But for now, sit back, relax. We are off on a journey to Lord Howe Island and a journey into history.
I saw a photo in the beautiful Australia part of the Woman's Weekly. I thought, geez, that looks pretty good. Paul's Pyramid, where the hell's that?
It's about 25 kilometres off Lord Howe Island. It's a mecca for rock climbers.
And Dave was up on Gannett Green and around the ledges.
That is extinct.
And that was all that was known, that there was one recently dead one found on Ball's Pyramid in 1964.
This species may still be alive on the pyramid.
There's been many moments where that sort of thing happens. You come down to one event that is make or break. That's why we do these things, for those sort of moments.
Instalment in just a minute, but we actually have the opportunity to speak with Asher, the filmmaker, and here he is right here. So I just wanted to say thank you so much, Asher, for taking us on that wonderful journey, and also for joining us from Bristol.
You're very welcome.
And I wanted to ask you what you're doing now.
So at the moment, I'm just working with the BBC in Bristol. I'm trying to make some more films, such as the one you've just seen. Fingers crossed.
Fantastic. Now, do we have any questions in the audience for Asher? Because he's just going to join us for a little bit now, and then he has to get off to go to work, because it's early morning in Bristol at the moment, so we don't want to hold him up too much longer. But is there a question from the audience? Go. Just there's a microphone coming to you, if you could speak into that, please.
I noticed the recent climbers' faces were grayed out in the sailboat. Are they surreptitious climbers, or is climbing allowed now?
Yeah, no, surreptitious is a good word for it. So yeah, they were. They took it upon themselves to climb the pyramid, so to speak.
we have another question? Okay, great. Just wait for the microphone, please, because then everyone will hear you, including the people who are watching online.
How many stick insects were found on Ball's Pyramid?
In that initial expedition, I think they only found about 10 or 20. So originally that's all they thought there was in that one bush.
Okay, thank you for your answer.
Not a problem.
I have a question. How did you get Patrick Honan to share the story about his underpants?
I didn't even know he was going to say that, to be honest. That was one he just surprised me with. But it was, one of those, what do you call it? It was just serendipity, I guess.
He's a very funny man.
Very happy when he said that.
I think one of the things that the film really does beautifully is have humour that's peppered all through it and have a light touch to it. Have you got any other sort of advice for people in terms of making very captivating natural history videos and films?
Oh, that's a tricky one. Well, this is very stylized in a way that I enjoy making films. So, personally, I tried to give it that nice mix of humor and heart and have some nice peaks and troughs, as it were, of jeopardy and release. So tension and release is something they say in storytelling theory.
Fantastic.
So
yes, we certainly got that. I think also, that whole thing about being right there in the action, whether it was actually on Ball's Pyramid or in looking after that one sick female, we were right there with the journey.
Excellent. That's good to hear.
And we got a last question for Asher before he heads off. Go for it. Yeah.
That's marvellous, Asher. Did you
Thank you.
really have to resort to crowdfunding to be able to make such a brilliant film?
Yes, is the answer to that. I had some very generous crowdfunders. The rest of it was basically from my own pocket. So it was very much a passion project for me. And it allowed me to just do a lot of those things I wanted to do, which potentially may have not made it into the film or in a different way.
We should make you an honorary Australian.
What was that? Sorry?
I think he is Are you an Australian?
I'm actually from New Zealand originally.
Okay. So you're getting honorary Australian status here.
Yeah. Thank you.
Fantastic. So thank you very much for starting us off on this amazing journey, and we hope you have a really good day there in the UK.
Yeah, you're very welcome, and thank you again very much for having me. I hope you all enjoyed the film, and thank you.
We're going to watch this spot for your next creative work. So let's
fingers crossed.
Let's give him a round.
Thank you.
Thank you. Fantastic. So to continue the story, we now have Paul Clemens, who's going to join us from the Australian Museum in Sydney, where he heads up their digital collections and citizen science, but he also has a particular role in their expeditions and the expeditions to Ball's Pyramid. So he's going to come on the line in just a minute. And when I asked him, when he was telling me about the expedition to Ball's Pyramid, he said that he was not a rock climber, so he didn't go perhaps as far as some of the other people up the very steep sides of Ball's Pyramid. But we'll hear more from him when he comes up online just in a minute. Is he? There we go. Hello, Paul.
Hi, Kate. How are you?
Good, thank you. Welcome. You're now in front of the live audience here, and
Very large.
We're looking forward to hearing what happened next. So.
Yes, well, let me share my screen so I can get started. I hope everybody can hear both myself, and I'm going to play a little video as well, not quite as spectacular as Ash's, but it's a great video as well. So I'll just share my screen. Okay.
Fantastic.
So as everybody hear me there now, Kate?
Yes, all loud and clear. That's great.
Excellent. So my name is Paul Clemens. I'm from the Australian Museum, and I coordinate expeditions run by the museum. The museum's had a long relationship with Lord Howe Island and the Lord Howe Island Board. And in 2017, as part of the Australian Museum's 190th anniversary celebration, the museum joined forces with the Lord Howe Island Board, Lord Howe Island Museum, New South Wales Office of Environment and Heritage, and the Melbourne Zoo, as well as the climbing community, to undertake a survey of Ball's Pyramid. The intention was to strengthen the Melbourne Zoo's breeding program and to support overall efforts to conserve the phasmid, particularly with the opportunity provided by the impending rat eradication on Lord Howe Island, which I'll speak briefly about a little bit later. A special approach to the Ball's Pyramid expedition was required due to the extreme nature of the collecting environment, which you have a bit of an idea of, and I'll show you more of in a minute. And so the museum engaged 6 expert climbers to access areas never before surveyed, and they were legally climbing Ball's Pyramid because as part of an organised scientific expedition, they were allowed to participate, and they offered their services for free, and all their skills, knowledge, and experience in climbing, which is very important, because the logistics and safety aspects of surveying Ball's Pyramid were at the very least extreme. It's not something that an institution like a museum or a government department would do normally. So it's really going out on a limb, you might say. It can only be accessed by swimming from a moored boat. So landing a team of 10, including scientists and climbers, and equipment for a stay of up to 10 nights was very challenging. So we actually stayed 9 nights on Ball's Pyramid. I'm just working out how to progress this. Okay, so I'm just going to play a film which is about the expedition, and we'll really give you an idea of what we did, what we went through and what we achieved, and then I'll talk to a few slides afterwards to tell you more about it.
Humans are curious creatures, and we want to understand the world that we are living in. How species are able to survive and how do they adapt to environments.
Ball's Pyramid is a unique environment, so we've come here to do a survey of the biodiversity of Ball's Pyramid and to look for the Lord Howe Island phasmid, found on only Ball's Pyramid, the only place in the world where it's found. The beauty of having the volunteers involved was that we were able to create something called extreme citizen science, where we had members of the community being involved in real science and understand better the biodiversity of Ball's Pyramid.
In this tube is some very precious cargo. We have Vanessa, who is our newest Lord Howe Island stick insect that's going to come back to Melbourne Zoo. We're hoping that they will be the stock that then come back to Lord Howe Island.
That film was made by Tom Bannigan. He was a contracted filmmaker that we took out with us on the expedition. So he made it on behalf of the Australian Museum. I just thought I'd go through a little bit of what it's like to run an expedition like that, I guess, and what's involved. And certainly from the very beginning, having those 6 climbers involved who had so much experience in climbing and who had actually several years before put in a proposal for carrying out an expedition to Ball's Pyramid. So we were very lucky to be able to some degree, right on the back of that, I guess, in terms of they'd put a lot of thought into what would be required to carry out an expedition to the island. So they very much hit the ground running. When I spoke to them, I spoke to Zane on the phone, and he said he had a team of 6 climbers who he could get in contact with. And they then set about the logistics, preparing all the equipment we were going to need. We needed to take out a lot of water. We needed to take into account it's very difficult to get on and off Ball's Pyramid, so we had to prepare for if we were unable to get off because of bad weather. So not only is it difficult to get on, but if the weather conditions aren't right, a boat won't come out from Lord Howe Island to pick you up. So you have to stay until the weather conditions improve. So we need to take out a lot of water, and we needed to take out plenty of provisions for food, and as well as the collecting equipment and tents, etc. Our sleeping equipment was pretty rudimentary. But before I go into that, actually, I just want to introduce you to the climbers. So these are the 6 climbers, and something you might notice about them is they're not terribly young. And when I first rang Zane, who's the second guy from the right, he's the youngest at 49 years old. I had no idea how old they were. And so I asked him to send me through details about the climbers, and he sent me through the details. And one of the first things that I noticed was the age of them all. And 2 of them, that's Keith Bell on the left-hand side and Brian Manick on the right-hand side, was 69 and 70 years old. And being a little bit risk-averse or concerned, I said to Zane, "You're kidding me, aren't you? These guys are pretty old." And so the next youngest, I think, from Zane was somebody in the early. So they're all over 50 except for Zane. And we had Brian and Keith, 69 and 70. So I said to, I think it was Keith I was talking to, I said, "Oh, well, what makes you guys applicable to this? I was expecting much younger people." And Keith quickly said, "Well, there's old climbers and there's bold climbers, but there's no old, bold climbers." The point being that if you were too bold in your climbing, you didn't last that long. So the indication that these guys were all at this age meant they were very careful and they were very considered about how their climbing went. So they were an incredible attribute to the project, was their experience and their wisdom and their very considered approach. We spent some time on Lord Howe Island before going out to Ball's Pyramid, training them up on how to look for eggs and droppings from the phasmids, and also we introduced them to what the phasmids look like from the Lord Howe Island. They have a very small breeding colony there as well. Now, just to give you an idea of where we stayed. So on the left-hand side there is a view from above of our tents, etc. And you'll notice they look very close to the waves and the ocean there. And as the expedition leader, I used to lay awake at night feeling the waves pounding just below us, and being somewhat terrified of being overwhelmed by the waves, because just before we went out, one of the expedition climbers informed me of a case where, in very similar circumstances, not where we were, but somewhere else, a change in weather had overwhelmed people that were staying in a similar situation, and somebody had been washed away and killed. So I was very nervous about staying for 9 nights in this location. It was very exposed. As you can see, there's no plants or anything around on the right-hand side there. They're actually our tents and our cooking area there underneath the tarp. And we had a generator, had all our water there. So we had to tie things down tightly. The weather wasn't great when we were there. It rained quite a bit, quite squally, and reasonably uncomfortable. And looking back now, I wish we'd taken out some chairs because we had to spend all our time sitting on those very sharp rocks, which became very uncomfortable. But the great part of this, where we were staying, was this is what you got to look at when you woke up every morning. And it's nothing short of spectacular, and really such an honour to have the opportunity to spend time out there. It's just a magical place. On these sort of trips, it's always great to have somebody that's going to provide you with fresh food. And this is Hank Bauer. He was from the Lord Howe Island board. He was with our base camp team, and he was quite adept at catching kingfish. And so on a number of days, we had fresh sashimi kingfish as part of our diet. The rest of the time, we were eating tin food, but that was a real treat to have fresh seafood like that. And Hank was a great provider in that sense. As you saw from the film, very extreme, well, for most of us, extreme conditions. You can see down there in the bottom, that's where Paul and Zane, who are brothers by the way, they slept in that cave there, which looks very tenuous to me, but I'm not that much of a climber. I did climb up about a third of the way up, but I didn't go all the way to the top like the other climbers who were doing the surveys. And for those of you that wondered what the view from the top of Ball's Pyramid was like, there's your 69 and your 70-year-old who climbed to the top, and that's the view from up on top of Ball's Pyramid. Nothing short of amazingly spectacular. And of course, that's Lord Howe Island in the distance, Mount Gower and Mount Litchfield. Now, just to give an idea of where we captured the phasmids from on Ball's Pyramid, there's a number of sort of landmarks that I've labelled on there. And the sightings of the phasmids you can see there, and those little white phasmids with a black background. So we surveyed up and down, right from the bottom to the top, and it was great to find that the phasmids were found right up that transect, and we expect would be found right around the pyramid, wherever the Melaleuca bushes were found. But we didn't find large numbers. To bring back one phasmid, we needed to find 10 or see 10. And so in the end, we were able to count 17 phasmids. So we're only able to bring back one, which we named Vanessa. We would like to have brought back more, but because of a recent drought, it had been quite a bad drought on Lord Howe Island and Ball's Pyramid, we think that had a big influence on the number of active and live phasmids we encountered. So what the expedition did demonstrate was that we had scientific proof that the phasmids were found spread all over Ball's Pyramid. So this is Vanessa. I thought I'd introduce you to Vanessa, who was the phasmid that came off Ball's Pyramid with us and went down to Melbourne Zoo for the breeding program. They're a beautiful looking animal, just glossy black, a bit like a patent leather sausage with legs, I guess. Now, moving on a little bit, I just want to talk about briefly a couple of things which is about really getting the phasmid back onto Lord Howe Island. There's a project being run out of the Australian National University by a chap called Alexander Mikhaev. That's the Lord Howe Island Phasmid Genome Project. So they're working on extracting the genome for the Lord Howe Island phasmid. They're using one of the phasmids from Melbourne Zoo, and they found that they've completed about 3/4 of the job, and they found that the genome is actually, for the phasmid, is larger than the human genome. Now, don't ask me how or why that is the case, but that is the case. I was talking to Alexander today. It'd be interesting to find more out about that some period down the track. They're looking at finishing sometime in the next year. And one of the major goals of the Genome Project is to explore the ancestral DNA structure of the different, I guess, specimens. We've got specimens here at the Australian Museum, the most specimens here. And so we've got a range of specimens from before before rats got onto the island. So we've got specimens of DNA can be extracted from for looking at their DNA from back then and looking how closely related they are to Ball's Pyramid phasmids that have been bred up at Melbourne Zoo. And the idea is to inform where the best source for phasmids, for DNA, sorry, for genetic diversity, I guess, for repopulating Lord Howe Island. So they're going to need to collect more phasmids from Ball's Pyramid to get a better understanding of how closely related they are to each other, but also to the phasmids in the breeding program at Melbourne Zoo. So that's a really important project for the future of getting the phasmids back onto Lord Howe Island.
Hello, Paul.
Now the other.
Are you just about to finish up, Paul? Because are you just about to finish up because we need to go on?
I'll make this quick because I don't have a lot that I can talk about the eradication project, but I can tell you that it's a really important aspect. Rats have led to the extinction of 5 bird species, 13 invertebrates, and 2 plant species that we know of on Lord Howe Island. And so the rat eradication program, which is underway at the moment and is the largest populated island to undertake a full-scale eradication of rodents. And it will be a key to reintroducing the phasmids back onto Lord Howe Island. If you want to know more about that, you can actually go onto the web and go to the Lord Howe Island website for rat eradication, and you can find out a lot more there about rat eradication, how they're doing it, and the likely outcomes there. So I'll leave it there. Thank you for giving me the opportunity to speak to you all tonight.
Fantastic. Thank you, Paul. And if you're happy to wait on the line, we'd love to save up some questions for after our next speaker, who is going to, a really nice segue, because I know that some of the things that he is going to talk about will extend your story. So thanks very much. And now I'd like to welcome Rowan Cleave from Zoos Victoria, who's going to talk to us about his work breeding the Lord Howe Island stick insect. And give us a feeling for the challenges involved with this particular species, and also, the links with the other groups that are doing captive breeding. So thanks very much, Rowan.
Good evening. Thanks to Melbourne Museum and in particular Zoos Victoria for allowing me to come and speak to you this evening. Normally, presentations I give will go for 1 to 2 hours, and I'm going to cut everything down really, really quickly tonight. I'll go through as much information as I can because this is a fascinating story of survival of a really unique species, and you've heard and seen a lot already tonight, so it's great to see the interest go through and on. So my name is Rowan. I've been working with the species since 2003. So I'm going to cover a lot of area tonight just on the species itself, captive breeding at Zoos Victoria, and we'll answer questions later on as well. So there's over 3,000 stick and leaf insect species in the world known, and about 150 species in Australia. Lord Howe Island is that red dot out there. It's about a 2-hour flight from Sydney. Beautiful spot. If you haven't been, I highly recommend it. To go and visit. It dates back to about 7 million years old and first settled and so forth. World Heritage listed as well. Population in 2016 was 382 and tourist numbers are limited to about 400 any one time. So it is a beautiful spot indeed. So the start of the phasmid story, I suppose, is an interesting one, and again, it's through accidental introduction of a species, and that is the black rat. This is the supply ship, the Macumbo, which ran aground, hit a reef off Lord Howe Island on the 15th of June 1918. It was careened closer to shore for 9 days, and in those 9 days, rats obviously got across and in a very, very short time wiped out, as Paul mentioned earlier as well, a number of species. So the red mark up there is where the wreck happened. Here are some early scientific drawings. These are obviously males, and I'll go into the differences between the males and females. It's very, very easy to see. These are the museum specimens out on Lord Howe Island, a male and a female. The female is on the left, your left, and the male is on the right. Very, very different features on both. Those two specimens are still out there. I was out there a couple of weeks ago, and they're still on display in the museum. So here are some basic facts about the species. They were listed as critically endangered and presumed extinct by about the 1930s. No one really knows the exact date, obviously, but mid-1920s, certainly by the 1930s. Rediscovered officially alive in 2001. Draft recovery plan was 2002, and in that time Patrick, who you saw in the movie, and another staff member, Norman and Ando, were working in the butterfly house at Melbourne Zoo and put our name forward to be part of the recovery plan as well. We have a long history of looking after invertebrates at Melbourne Zoo with the butterfly house. So captive management of the species commenced in 2003. 4 individuals were removed from Ball's Pyramid. 2 of those came to Melbourne Zoo and they were named Adam and Eve, and 2 went to an entomologist, Stephen Fellenberg, who was also credited with rediscovery of the species in 2001. His pair he took back there as well. So inhabit forests. Adults, about the females are a lot longer and bigger than the males. Lifespan of about 2 years and over. Very dependent on what they do eat as well, with lifespan and longevity. The eggs hatch between 6 and 9 months. Just one thing I want you to think of is that all this information I'm telling you tonight, when we first became caretakers of this species, we knew absolutely nothing about them except the plant that they fed on, that's it. So the nymphs are about 2 centimetres long and they come out bright green, and they're 3 times the length of the egg, and there's a slide coming up that I'll show you that distinct difference as well. So they weigh up to about 25 grams, which is about a tablespoon of butter. The adults are nocturnal. The young are not so much diurnal, but they sit out in the light during the day very, very comfortably. They'll molt. That molting process can happen day or night, particularly in the young ones. It'll happen during the day, and they'll move around. They'll drink off-leaf material if you give them water to drink. And they have some great names: land lobster, tree lobster, and walking sausage, as you heard earlier as well. So at the time of the rediscovery of live specimens, there were 3 found in 2001. So Ball's Pyramid, I don't need to go into a lot of information there, but it's absolutely spectacular. Seeing it from Lord Howe Island itself, it's so impressive. So it is a fragile environment, which I think you've probably heard this evening as well, and disturbance from climbers, scientists, weather events in particular, collectors, landslides are a real concern out there. One real big issue is probably weed invasion, and we're great support from the Lord Howe Island Board weed team there of clearing the coastal morning glory, which does take over and can take over the Melaleuca howiana bushes very, very quickly indeed. And obviously again, the arrival of rats or mice from sea or boats or floating vegetation. So these again are some of the early photographs some of you saw earlier, and it is not a very pleasant place obviously to get on and climb around and be on. Very, very dangerous. Here are some of the early photographs of that expedition and the Melaleuca howiana bush here, the big one that was also mentioned. Here are some of the approved survey results. So you can see it does fluctuate quite a bit with numbers. So 2 adult females, 1 nymph was the first rediscovery, and then 24, 25 and 5 nymphs. So it's a very, very small population, and the concern over the past couple of years over summers has been they've been very dry summers, and there has not been a lot of specimens seen when there has been expeditions out there, and there hasn't been a lot of frass seen either under the bushes. So it's a real concern for the population out there. So Melbourne Zoo, hopefully you all know this place and know what it does. The Butterfly House is where I work, and behind the scenes, these were some of the earlier glass houses that we took over for this captive breeding program. These ones have now been replaced to more modern, updated facilities. And the photographs of Adam and Eve, so these were obviously night photographs taken by Patrick in full darkness, male on the left and the female on the right, Eve. So again, once that started, we're learning things constantly in that time, and going from letting animals walk around a glass house completely to then putting them into an enclosure. This is actually the first generation from Adam and Eve, some of their offspring for breeding. So there's plants in there, there's nest boxes, there's egg-laying containers. The females will actually deposit their egg under the ground. There's lots of little hides and things. They are great at trying to escape, so we do make sure that everything is pretty secure. And we have modified things completely over the years. Eve's story is a really interesting one and very emotive and really connected with a lot of people. Where she was very, very ill, as Patrick mentioned in that movie, and she was upside down with her legs twitching. So he actually made a concoction of Melaleuca howeana leaves that he ground up, glucose, calcium and distilled water in consultation with our vets and worldwide research. So she recovered, and you can see the eggs there quite easily, 6 eggs in her abdomen from that x-ray, and just out of interest, none of those eggs were viable post-x-ray. She went on to lay 248 eggs, and obviously the cornerstone of our captive breeding population at the zoo. So the first nymph to emerge, and again we were going in every single day checking the eggs. Again, you've got no idea when, how long they're going to take to hatch. And we were really surprised that the nymph was bright green. That's my fingertip there. That is not actually the first one. That's just showing the size of them. It's about 2 centimetres. And to come out on Threatened Species Day, it was just, you know, an amazing story, and we promote that story on Threatened Species Day as well. So a number of glass houses have been built, and this is just flicking through them. Some of them are next to the Butterfly House, some are behind, and they're all in off-limit facilities. So highly restricted. This one here in particular was designed and built with keeping staff in mind, with our input into what needs to go into saving a species, and that can be used for other species in the future as well. This is a higher quarantine facility here with an airlock as well that's open and has Vanessa's offspring, the female phasmid, collected in 2017. So here's just some general enclosures. So we raise the young in enclosures that years ago we built them ourselves, but now we're buying stock off the shelf that operate just perfectly for this and our program of raising young. So in an enclosure like that, we'll put about 30 nymphs and we'll raise them up till about 3 months of age, and then put them into a free-ranging glass house, and I'll show that later as well. Anything with sub-adults in them, we provide what we'd call nesting boxes, and they again have been modified. There are some really important things in that photograph, and one is at the front on the right is the sand tray. It's just sand and peat for egg laying for the female to have access into, and a petri dish for water. They will come down and they will drink water. They'll put their whole, pretty much most of their mouth parts under the water, and you can actually see them drinking as well. Ficus there. So since 2013 and prior, we've been giving them plant choice as well, which is really important. And we're going to more and more endemic plants off Lord Howe Island as well, and thanks to the board out there and the team were able to collect seeds for us when we grow and propagate at the zoo. So this is what we would call one of our free-ranging, so there's sub-adults and adults. There's sub-adult at about 3 months, 2 to 3 months of age, and adult at around 6 months of age, lifespan of 2 years I mentioned earlier. So within that environment, they have full rein to come out overnight. About 2 hours after dark, they'll be out, and they'll use that whole glass house. They'll be over the sides of the walls, up on the roof. Most of them obviously will be on the ficus there. Ficus is their favourite plant, and we've done a lot of plant trials over the years. The other one here is tree lucerne in the foreground. So we keep things really clean within those environments. We've lifted the boxes up off the ground to replicate trees. I move things around once a week, keeping them as active as possible. And again, those boxes have been designed by us and made internally at Zoos Vic, and they're just on an angle. They do actually like to go in and up to a higher level. It's obviously darker up there, and all their frass, their poo and eggs roll down the box and out down the bottom. There's a little slit down the bottom. So the underside of the adult male and female. Female has this beautiful, very sharp, it's not so much sharp, but it's very hard ovipositor. And she will actually dig that down through the soil, deposit the egg, come out, smooth over and walk away, one egg at a time only, and the male there. Mating can occur for quite a few hours as well overnight, and you'll only see them mating at night. The males, this is a very, very impressive male we've had bred at the zoo in 2012. I named him Sir David in the end, because he was the male that got to meet Sir David, obviously. He is very, very impressive male that came through. So egg laying, they will just have eggs come out just above the ovipositor, and this is a female bearing down. You can see how far she does go down, and again we measured all that distance right from the start, and we were burying the eggs at the same distance down in the substrate as well. So the egg management, eggs are collected once a week. A sampling of those eggs I get out and we'll measure length, width and weight. There's about either 10 or 20 eggs a week we do. So in total there's about 40 eggs that will be measured and then set up. That's a pretty typical weight of the egg, set up in a little plastic container in vermiculite and buried. So the eggs, we always keep them buried. We've found that if we keep them out just by themselves, they become non-viable very quickly. The yolk inside dries out. So for a very short period of time, probably a year or so, I was numbering eggs. So we had direct correlation of length, width and weight to the nymph that emerged. So it's a lot of work that goes into it, but a lot of science behind it as well, which is really interesting. So this is just a little setup I've got at work. I go in and measure them up, record everything onto data sheets. The eggs go into rows like this. They're kept moist, so once a week, once a fortnight, they may get a very light mist spraying, and then that promotes also hatching. So we have lots of data on those as well. So here's an egg, and you can watch one hatch online. There's a hatching video I took many, many years ago now. That is online. You can access that if you'd like to go look at that as well. And again, this was for our team, staff and people to connect with the species. That's been part of my job as well, as trying to get people to connect to the species. This is a great story that should and needs to be told about conservation programs and what can be done with very, very small numbers of staff. And a commitment from an organisation. So that's the size. So the nymph that comes out is 3 times the length of the egg. And some record keeping is really important. So we went back through and just started writing everything down. And since the start of the program, over 16,000, and I'll show that pretty soon, nymphs have hatched at the zoo. So all the data goes into ZIMS. It's called a record-keeping system. It's online. So anyone that works in a zoo or organisation where you have animals, you might be using this system. You know, and I can do thousands of entries in a month. And this is sort of the data that's collected. All that information can be put into papers and so forth. And hopefully that'll come in the future with staff doing it. We work with a lot of great master's students as well. So here's the hatching numbers at Melbourne Zoo alone. So you can see 2003, 2004, 2005 were very, very stressful times for the program. we were operating in such small numbers hatching. And then 2006, things just kind of clicked. We learnt a lot, and from then on, when there are huge numbers, probably the 2000 plus, we were doing a lot of research projects with master students from Melbourne Uni and so forth. So you can see there we've just clicked over 16,500 nymphs at the zoo. So this is what they come out like. They'll come out into those containers. The nymphs will always go up on top. Every single nymph that's ever hatched at the zoo has had a measurement from tip of the head to the tip of the abdomen, and that's a pretty good measurement again. And then they're raised up at the zoo. So this process, molting process, really beautiful one. Again, you can see this vision online. The process itself for the young ones can be about 10 minutes. Anything, the sub-adults going into adult can take up to about 20 to 30 minutes. It is a slow process, but yeah, it is a beautiful one to watch as well. They'll hang there. The nymphs can actually turn around and eat the old exoskeleton that they've just come out of. And here's some photos of the young. You can see them changing colour. So they go from green to green-brown to brown. And then here's a sub-adult male, and there's this nice little split, and then the pulsing out comes out. And it'll take about 24 to 48 hours for their exoskeleton to harden completely again before they go that jet black. So adults, the male and the female, very obvious there, those two to see, and that's the male there on Melaleuca howiana. Head profiles, a lot of people kind of find these really freaky, but they are cool to see. See the mouth parts there, and that one's very alien-like as well. They're really cool. So the first month or so, they're sort of by themselves, and then they start this clumping behaviour, and they keep that behaviour up, very, very communal. And then if you have a pair together, this is the behaviour that you'll see. The male will usually be on top of the female with his legs over her body. We grow a lot of plants at the zoo. Our nursery team are absolutely incredible for us. Propagation of plants, endemic plants of Lord Howe Island, and this is the plant nursery at the zoo. We also do, obviously, they're getting plants for the butterflies for us, the caterpillars. This is a very small sample. So we do a lot of investigations into deaths. There's a lot of research going into invertebrate medicine at the moment, and Paul mentioned earlier about the genetic research. We're doing also DNA extraction techniques of frass and all sorts of stuff. There's a lot of information to come in the future of that. They can reproduce parthenogenically, so they don't need males to reproduce. I ran 3 generations through of parthenogenic only trial, and we did some overnight studying. We're now in our 14th generation, probably another few months, and hopefully we'll get to our 15th at the zoo, and this just indicates group 1. So we can track that group all the way back to Adam and Eve. And research master student Hayley, who's in the audience tonight, worked with us doing research back in 2012, and then we did some diet research on Lord Howe Island with another master student in 2013. And this was all building, do a lot of presentations out on Lord Howe Island again, community involvement. This is the board offices enclosure out on Lord Howe Island. So we sent animals back in 2008, 2009, and they've been keeping them there successfully. And you saw in the movie we've been given permission and approval to have an international breeding program. So very proud of Melbourne Museum, the work that they do here, also Toronto Zoo, Bristol Zoo in particular, and San Diego Zoo. So we're hoping to send animals again soon with that. This is the current enclosure on Lord Howe Island where they keep them all. So this is just a glass house again, designed specifically for them. This is the nursery. Probably the most important piece of equipment in there is this one here, humidifier. Environmental conditions are extremely susceptible to change. So if there is a breakdown, if there is an issue, that's when we have problems. So we've had a lot of worldwide recognition for the program. Little things have popped up all over the place in some major places all over the world. There's huge interest in this species and the program, and it is, as Patrick mentioned in the video, it is highly regarded in the conservation field, particularly invertebrates, but it's just in general of what can be done, you know, making the New York Times, for example. Dr. Jane Goodall and Sir David Attenborough both have expressed an interest in coming to Zoos Victoria to see the species. Both were really, really keen to get involved, come and see them. Declare your love for the phasmid. This is one of our programs that we had at Zoos Victoria. Anyone else that's really interested, Asher did crowdfunding. Gilly Rose, who was an animator, again approached me about making an animation movie. You can actually access this one online too. Again, highly emotive, beautiful animation movie that circulated the world and promoted the species, which was something lovely. And that's Gilly there. She's actually overseas at the moment. Mr. Rick Wilkinson, who wrote this book, Return of the Phasmid. Heaps of information. Rick's here tonight. But this is a great book if you want to delve deeper into the people behind the history of Lord Howe Island and so forth. Asher, as you just saw, and it's the first species to have a species management plan. So every year I have to write a report on the species and numbers and so forth. Vanessa, who obviously spoke about earlier as well, so you may or may not have made that connection that the climber Vanessa, this animal was named after her in her honour, and we have her second generation being bred and being raised at Zoos Victoria at the moment. So there's some of her information now. Don't need to go into too much information on this, but rodent eradication has started on Lord Howe Island, 22nd of May that started, in an effort to get rid of black rats off Lord Howe Island. So anything from hand baiting stations to helicopter aerial baiting stations, depending on where they are on the map there. Here's just some of the grid. The little boxes there, you can see they're there because they have cattle in that area, so cattle won't stand on little pyramid-shaped boxes. So they're grid 10 by 10 metres and checked constantly. So that's just the bait stations there. And this is Blackburn Island, which is just across the lagoon. That's a big ficus out there. And we, along with the Lord Howe Island Board, conservation volunteers, the Friends of Lord Howe Island and visitors, have all got involved in that program to revegetate Blackburn Island. And that's seen as a potential soft release site for the species as well into the future. And yeah, we'll see what happens. The future is looking much brighter for the species. Thank you. And that'll wrap up from me, but happy to answer questions as we go. Thank you.
Thank you so much, Rowan. That was fascinating. We do have a little bit of time for questions. I know we're running up perhaps slightly behind schedule, but it'd be great to hear a couple of questions from the floor. And you can either ask Rowan or we still have Paul from Sydney.
Okay,
from
either one, I guess. So kind of two questions. One, can you track individuals once they're in these group enclosures? And the second is, do you allow them to find their mates on their own, or you put them into a pair?
Yep, so we don't track individuals unless we need to. We have in the past for research projects. We've split populations in the past. We have tracked individuals. We can track different groups. If we need to track an individual like Vanessa, we obviously do, and all that information goes into her code online into the ZIM system. Early on, we were doing our best case scenario with regards to captive management breeding. So we had the individuals separated in different enclosures, and every couple of weeks we were rotating males through. It wasn't until we were able to swap animals with Stephen Fellenberg's population and then start breeding really successfully that we were able to go to we felt that we were able to go to a free range situation. So now our populations in particular, they choose their own mate. So at that point when we're able to go to that, the egg size became bigger, the nymphs became stronger and so forth. So it's, yeah, and that continues to this date.
Do you get a feeling from looking at animals in captivity how mobile they would be on the pyramid and how far they would be able to range naturally?
Yeah, absolutely. They're very, very mobile. They will cover as much area in our environment, so I have no reason why they wouldn't on Ball's Pyramid. And you can see by the footage that Paul showed where the numbers were found as well, that they are all over.
The potential soft re-release on Blackburn Island, two-part question. Is there any records of Lord Howe Island stick insects being there before? And are there rats there? Is that part of the rat eradication problem, or did rats not reach there?
Yeah, I don't think they've ever been there before. And even though there has been no rats seen on Blackburn Island, we know that that was baited as well. So I went out to Blackburn Island when we were there, we were planting, and there were the signs around that they put up everywhere that rodenticide had been placed out there.
Could I just ask about the cattle that's on Lord Howe Island? Has that ever had an impact on the phasmids?
No, I wouldn't think so. Not that we're aware of, no.
Cool.
They would have come in. They would have come in after.
Was there a question somewhere down here? Yeah.
Thank you. I was wondering, with regard to the reintroduction program, what is the outlook regarding the homogeneity of captive individuals? Because obviously you've got a very small gene pool. Is that a potential risk?
Yeah, so there's a team being put together now. Obviously that works very, very closely. Zoos Victoria, the museum, Lord Howe Island Board are all involved, and the New South Wales OHH, the Office of Environment and Heritage, I think they're called something else now, but there's a team put together to discuss all of that. Now there are a lot of different scenarios that could go and play out. Obviously, firstly we need to find out that there are no rodents on Lord Howe Island anymore. So that's potentially a 2-year process before anything is considered with regards to going back. Now there are a number of different plans. We're doing a lot of disease risk analysis now at Zoos Victoria with our vets and veterinary team that is involved in that currently. So a lot of that data isn't available yet and research is ongoing. But you know, the likelihood is that there are a number of scenarios that still could play out, quite possibly.
Do we have a last question? Okay, or maybe two, as long as they're quick.
Any plans to go back to Bells and find another female?
Maybe that's one for you, Paul. Did you hear that question?
Yeah, I heard the question. There's no concrete plans that I'm aware of, but I do know that Lord Howe Island Board has expressed interest. I mean, ever since we came back from Ball's Pyramid 2 years ago and we brought Vanessa back, there's been interest in going again and getting, they wouldn't be such long expeditions though, they're more likely to, now that they've got a bit of an idea where to go, to get additional individuals and how to go about doing that, they'd probably go for a couple of days at a time rather than 9 days. But no concrete plans that I'm aware of yet.
Is it giving you nightmares thinking about it, or would you contemplate it?
Oh, I'd love to go. I'd go at the drop of a hat. It's a magnificent place and a very special place, and I'd love to go again. It's very uncomfortable to sleep on that rock, but it's a very beautiful place.
Okay. Okay, last one, thanks.
Yes, my question is about Blackburn Island. I'm surprised you're selecting that as a place to reintroduce the animal, because it's a pretty dry island and it's pretty exposed. I would have thought it was just the wrong environment for the stick insect.
It's an option, and at the moment, all options are on the table. So it's the one good thing about that place, it has that beautiful large ficus tree that you saw there. So re-veg is happening. It is a potential soft release site as a trial site to see how things go, and then the potential back onto Lord Howe Island. And as I mentioned earlier, there's so many things yet a lot of planning still going into all of that.
Well, thank you, everybody. I'd like to join with me in thanking our speakers, Paul from Sydney and Rowan from Melbourne. And I'd just like to invite you along to come to our next lectures. Also, just mention this book, which Rowan Cleve wrote with Coral Tulloch, and it has just won this Environment Award for Children's Literature here. So well done, congratulations. If you want to get a copy of that, it is available at our shop, which is now closed, but you pop back another time, and I'm sure it's available from lots of other good bookstores. Do join us for one of the other upcoming lectures. Our next one in October is Anarchy in the Archive, the protest collection at Museums Victoria. And then in November, we have Stardust Memories, 50 Years of the Murchison Meteorite. So thank you very much for coming along tonight, and I hope to see you here again soon.